Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics. This is plant structure and function. Going to topic 4A1, which is the cell wall. The plant cell wall gives the plant shape due to turgidity of the cells, meaning they take up water by osmosis in order to remain turgid. It also gives strength as well as support because there is lignification that takes place. The cell wall is mostly made up of insoluble cellulose. And then the plant cell wall, this is usually freely permeable to everything that is dissolved in water. Sometimes lignin is going to be added, like in woody plants, as well as suberin in cell walls of cork tissue, and this is going to reduce permeability to water. Moving on to layers of the plant cell wall, there is the middle lamella, which is the first layer, and this contains pectin, which is a polysaccharide. It acts as glue, holding the cell walls of adjacent plant cells together. If I can show you in this image, we can see this is the middle lamella and it's present in between the cell walls of different cells. Pectin binds to calcium in order to produce calcium pectate, which we're going to find in the middle lamella. In cellulose cell walls, there are microfibules, and these build up on both sides of the middle lamella. And again, if we use this same diagram, we can see this is the middle lamella, that side and the other side. And then there is the primary cell wall as well as the secondary cell wall on either side. And here, we can see the primary cell wall and then the secondary cell wall, which is going to be this one here. This is the secondary cell wall. That was the primary cell wall. In this case, this is the primary over here as the secondary is formed beneath that. The secondary cell wall builds up with cellulose microfibules, which are densely packed at different angles to each other. The cellulose microfibules are going to be laid up in layers. However, those in each layer are going to be at different angles or right angles to the ones in the different layer. There is going to be hemicelluloses that harden it, and then lignin is added to the cell walls of woody plants. There is no lignification that occurs in the primary cell walls. As you can see, lignin is only found in the secondary cell wall. In the primary cell wall, we have only hemicellulose. Moving on to the chemistry and structure of cellulose. Cellulose is the main component found in plant cell walls, and it contains only of beta-glucose molecules, which is joined by 1,4-glycosidic bonds. In cellulose, which is a polymer, one monomer unit has to be turned over so that the bonding can take place. The turning over I'm talking about is this one here. There is inversion. So the first glucose molecule is going to be upright while the other is going to be inverted in order to produce the glycosidic bond. During the inversion, the hydroxyl groups are going to stick out on both sides, and this allows cross-linking between them as hydrogen bonds can form. The cross-linking makes the material with great strength, so it's going to be used for strength as well as supporting plants. The cellulose molecules do not coil or spiral like we see in starch. They remain as very long straight chains, so they are useful for support. Moving on to the plasmodesmatter matter, which is the region we see here. And of course, around the plasmodesmatter matter, there is no secondary thickening, so it means only part of the primary cell wall. Materials are going to be exchanged through this plasmodesmatter matter, which are cytoplasmic bridges. Around these regions, the cells are thinner. During cytokinesis, two daughter cells will not completely separate, as threads of the cytoplasm remain between them. The interconnected cytoplasm are called symplast. When secondary thickening occurs, the hemicelluloses and lignin are deposited in the cell wall, making it harder. However, this is not going to occur in the regions of the plasmodesmatter matter, and therefore the pits are going to remain. These pits allow for water movement between cells, especially within the xylem. They're important in maintaining a flow of water at even pressure throughout the plant. Moving on to topic 4A2, plant organelles. The first is a permanent vacuole. A vacuole is a fluid-filled space within the cytoplasm and is surrounded by a membrane called the tonoplast. You can see that. The tonoplast controls the movement of substances in and out of the vacuole, and it also controls the water potential of a specific cell. Vacuoles contain cell sap, as well as other substances and pigments. In controlling the water potential of the cell, the vacuole contains dissolved solids that allow water to move into the cell by osmosis in order to cause the cell to become turgid or swollen so that the plant can remain upright. Next, we go to the chloroplasts. These are found in cells from green parts of a plant, and they enable photosynthesis to occur. Cells found in flowers, seeds, roots, transport tissues, and internal tissues of the stem have no chloroplast because they're not going to be exposed to sunlight, and therefore there is no photosynthesis taking place in there. You need to remember the structure of the chloroplast. It contains the stroma, which is kind of the internal space. There is the grana, which is the stack of thylakoids. And again, we can see the thylakoids here. There is the inner membrane, as well as an outer membrane. It's an organelle, so it's membrane bound. There is the stroma lamellae. There is the intermembrane space, the space between the two membranes, and then the lumen. Please practice drawing and labeling the chloroplast. Moving on to similarities of chloroplast and mitochondria. Both are organelles that have a biconcave shape. They contain their own DNA. They are surrounded by an outer membrane, and they have enormously folded inner membranes. These foldings provide large surface area for enzyme-controlled reactions that occur within the inner membrane, and they are thought to have been free-living prokaryotic organisms that were engulfed and became part of cells more than 2,000 million years ago, and this is through the endosymbiosis theory. However, chloroplasts do contain chlorophyll, but mitochondria do not. Chlorophyll is a green pigment which traps sunlight in order for photosynthesis to occur. The next are amyloplasts. They are colorless organelles that store starch. Starch can be converted to glucose in order to provide energy that the cell needs, and large numbers of amyloplasts are found in storage areas within the plants, for example, potato tubers. So this is an example of a plant cell, and we can see the amyloplasts are in here. They contain starch granules, and starch is going to be stored in there. Moving on to topic 4A3, plant stems. Plant stems function by giving the plant support to hold the leaves in the best position for photosynthesis to occur. They also support flowers to maximize the likelihood of pollination. They also allow for movement of substances around the plant, meaning they provide a route through which the products of photosynthesis are carried up and down a plant. And then they allow for movement of water and dissolved ions from the roots towards the leaves. Plant tissues we're going to look at include the xylem, the phloem, the sclerenchyma, collenchyma, as well as the parenchyma. We can see them in this image. We have the vesicular bundle here, and this is the sclerenchyma, which is going to be found around all the vesicular bundles. And then we have this, which is the collenchyma, which is for support. We also have the xylem as well as phloem, and these make the vesicular bundles. We have the cambium, which is unspecialized cells that can make the xylem as well as the phloem. And then we have the parenchyma, which are the packing tissues. We're going to look at each one at a time. Beginning with the parenchyma, these are unspecialized cells that can be modified for storage as well as for photosynthesis. They can also be modified to produce collenchyma as well as parenchyma. They give buoyancy to aquatic plants, and we can see this is how they look, the longitudinal as well as the cross-section. Next we go to the collenchyma. These have thick cellulose cell walls, but these cell walls are primary cell walls, so there is no lignification, and they are thicker at their corners. We can see the thickening that occurs at the corners. These are found around the outside of the stem, like we already saw in the other figure, and then they give plenty of support but remain living, so they can stretch as the plant grows to provide flexibility. Sclerenchyma are modified from parenchyma, which were the packing tissues we saw already. They are found around vesicular bundles of older stems as well as leaves. Or sclerenchyma cells have secondary cell walls, meaning there is going to be lignification. Lignin is deposited in the cell walls of the fibers in spiral as well as ring pattern, and this makes the fibers very strong, but they retain their flexibility. Sclerenchyma cells can completely become impregnated with lignin in order to form sclerates. Moving on to the transport tissues, which are the xylem as well as the phloem. Xylem tissues carry water and dissolved minerals from the roots towards the upper parts of the plant so that photosynthesis can occur as well as cooling. Phloem are living tissues. They are made up of phloem cells, and these transport the product of photosynthesis. This should be photosynthesis, which is mainly sucrose, from the leaves to where they are needed for growth or to be stored as starch. The cambium is a layer of unspecialized cells which can divide to give rise to xylem as well as phloem. So we can see all this here. We have the phloem, we have the xylem, as well as the cambium in here, and the sclerenchyma are going to be on the outer part of the vesicular bundles. The xylem usually start off as living tissues, and we call these the protoxylem. They can stretch and grow because their cell walls are not yet lignified. However, as they grow, more lignin is incorporated into their cell walls, and then they can stop growing. Then they become impermeable to water and other substances. These lignified tissues are called the metaxylem. Their end walls are going to break down, creating hollow tubes, which go from roots to the tip of the stems as well as leaves, and then water and mineral ions can be transported through them. Water can move out of the xylem to the surrounding cells through the pits, which we already saw. This is what we see formation of the xylem. We can see the cells are originally living. However, due to added lignin, they die. The cytoplasm is going to be lost, and then the transverse plate is going to break down. When it breaks down, we create hollow tubes that can transport water and mineral ions from the roots towards the leaves. Moving on to the phloem is never lignified, so it's living tissue, and it's where the transport of organic substances like sucrose occurs up and down the plant, and this translocation process requires ATP because it's an active process. Phloem consists of many cells. These are going to be joined to produce very long tubes from the highest shoot to the end of the root. The walls between these cells are going to be perforated, creating what we call sieve plates, and these sieve plates are going to allow the movement of sap through them. Mature phloem have no nucleus, however, because they're associated with companion cells enable the phloem cells to function, so we say they are still living tissue. And the phloem cells are going to be linked to the companion cells through the plasmodesmatter matter. So we can see here, these are the sieve tubes, and then we see the sieve plate. We see the companion cell that is living, it has mitochondria, it can produce energy and so on, so it can provide energy for this active process for movement of substances through the phloem tubes. Moving on to topic 4A4, the importance of water and minerals in plants. Water is needed for photosynthesis. It's needed for support, meaning turgidity, which maintains the cell structure as well as the plant structure. Water is needed for transport, for ions as well as organic molecules. And water is needed for cooling because evaporation occurs after transpiration, and this leads to cooling. Then plants need mineral ions like nitrates are needed to make amino acids, which can be used to make proteins, as well as the making of nucleotides. If there is deficiency, this can lead to stunted growth as well as yellow leaves. We can see that here. Calcium, this is needed to make calcium pectate, which is found in the medullamellae. We can say plural medullamellae. And deficiency of calcium leads to yellow crinkly leaves. We can see them here. Also, magnesium is needed to make chlorophyll. If there is deficiency, chlorophyll cannot be made. Photosynthesis is not going to occur. There is going to be slow growth. The leaves are going to be like that. They're not green because there is no chlorophyll. Moving on to topic 4A5, using plant starch and fibers. Plants contain fiber in the cellulose cell walls. These are usually long sclerenchyma cells or xylem tissues. They are very tough and strong, so cannot easily be broken down by pulling. We can see the different plant fibers. There is hemp, there is jute, manila, flax, and sisal. These are some of the products that can be made from plant fibers. Plant fibers can be processed. These fibers are processed by two methods. There was a traditional method which relied on natural decomposers, whereby the material was put somewhere and then decomposition occurred, and then the decomposed parts were washed off and the fibers were retained. Or we can use the manufacture method, which is more modern, in which chemicals or enzymes are used to decompose away the easily decomposable stuff leaving behind the fibers that cannot be broken down. After the decomposition, the remaining material can be spun into fibers. Spinning creates long continuous threads from short fibers. Still looking at plants, we can look at wood. Wood contains lignified cellulose fibers, which are embedded with hemicelluloses as well as lignin. Cellulose makes the wood resistant to compression. Also, wood contains a flexible matrix with intermeshing cellulose fibers, so nails can be hammered through without cracking. Moving on to bioplastics and biological polymers, they can ask you about the benefits of using biological polymers in comparison to using synthetic polymers. Like we already know, most plastics are made out of fossil fuels, but these cannot be sustainable because they are not renewable. However, bioplastics can be sustainable because they are made from renewable resources like sugarcane as well as maize. These bioplastics are also biodegradable, meaning they can be broken down by microorganisms. Bioplastics can be used for drug delivery, like in capsules. They can be used for drinking cups, computer casing, and so many more. And when these biological plastics are burned, they do not increase the net carbon dioxide concentration in the atmosphere because the CO2 released when you burn them is almost equivalent to the CO2 they took up during photosynthesis, so we can say they are closer to being carbon neutral. Moving on to topic 4A6, plant-based medicines. plants can fight some microbes on their own. These include bacteria as well as fungi that can infect plants. So this means the plants can produce chemicals which are antimicrobials. So we can use some of these to fight against human infections. In order to test some of the biological molecules got from plants, we can carry out bacterial culture to see how effective they are. In carrying out bacterial culture, we need to provide the right temperature, the correct nutrients, the right pH, and some bacteria require oxygen to grow if they are aerobic. Also, when we culture bacteria, we have to use aseptic techniques, and this is in order to prevent contamination from other bacteria, to prevent the growth of pathogenic bacteria. These could affect you or the person who's working on the experiment. Also, care has to be taken because non-pathogenic bacteria can mutate during the culture, and these can become pathogenic, so we have to be very careful. Also, we need to decide which growth medium we're going to use. Are we going to use a solid medium or we're going to use a liquid medium? For example, you can see this is a solid medium and there is bacteria already grown there, as well as a liquid medium. During bacterial culture on a solid medium, like agar, we will dip the sterilized loop into the bacteria suspension. This loop is going to be sterilized by flaming it. You flame it red hot and then allow it to cool. Then you will dip it into the bacteria suspension and then strike the agar surface. Like here, they are striking the agar surface. Then you can replace the lid and then put tape and label the petri dish and then turn it upside down. Turning it upside down prevents the condensed vapor from dripping onto the agar. If you want to determine antimicrobial effect of some plant extract, dip the card filter paper into the antimicrobial agent, and then you'll use sterile forceps to place the soaked filter paper onto the inoculated petri dish. You can culture for 24 hours or more, and then measure the diameter of the zone of inhibition, or we call it the diameter of the clear zone around the filter paper. The antimicrobial agent with a wider diameter of the clear zone is going to be the most effective. Looking at antimicrobial plant extracts, we know plants produce antiseptic compounds. Of course, many drugs used against infections in humans have been extracted out of plants. The extraction is carried out so that the dosage can be controlled, because if people just consume plant products without knowing the concentration, then the dosage cannot be managed. To extract the plant antimicrobials, we use a suitable solvent. The solvent must be able to extract as much compound as possible, meaning to maximize the extraction. The solvent could do this by breaking the membranes, this could be the vacuoles as well as the cell membranes, to cause leakage of the compounds out of the cells. During this process, grinding, mashing, cutting, and so on could occur in order to maximize the extraction process. So these are some of the methods or ways in which some of these are extracted out. Going on to topic 4A7, developing new drugs. Looking at testing promising new drugs, the new drug should be effective, meaning it should cure, prevent, or relieve symptoms of a specific disease. It should be safe, meaning not toxic to the body, and it should not cause unacceptable side effects. It should be stable, meaning it should not decompose under normal conditions. It should also be easily taken in and excreted from the body once it has done its job. It should be easily made on large scale and cheaply. In order to determine which drugs can be successful, computer models can be used for structures on active sites of enzymes as well as receptors of specific proteins that are involved in the progression of disease. This is for scientists to find out which compounds could be helpful. Once certain compounds, either binding at receptors or binding at the active sites of enzymes, have been discovered, they can be patented, and the patent can be up to 20 years. Testing can begin with cell cultures, tissues, and then whole organs in the lab before transitioning to animals as well as to humans. Drug testing can be carried out on animals in order to determine a good delivery system. Are we going to use a tablet? Is it going to be liquid medicine? Are they going to be injections, nasal sprays, and so on? And of course, the drug should be stable, meaning it should not break down into something toxic. And if the drug is changed into something else in the body, that should be safely excreted. Also, due to ethical concerns, animal trials can be replaced by tissue cultures, although animal trials must be carried out before moving on to human trials. So this is about minimizing the number of animals that are used in the trial, although we must carry out some trials on animals before we move on to human trials. Human clinical trials are carried out in 3 phases. There is phase 1. This is carried out on healthy volunteers, and this is to find out if there are any unknown side effects of this medicine to the human body. There is phase 2 clinical trial. This is carried out on about 100 to 500 patient volunteers, and this is to determine the ideal dosage, effectiveness, and if there are any side effects as well. Then in phase 3, this can be carried out on thousands of patient volunteers, and this is to confirm the effectiveness as well as the safety of the new drug. Also, this is to monitor if there is a statistically significant difference between the new drug and those that are currently available. Both phase 2 and phase 3 of the clinical trials are going to be double-blind, meaning the doctor, the scientist, and the patient do not know who gets the actual drug or who gets the placebo. The placebo, this is going to be given in order to eliminate biased results. Of course, a placebo is something that does not contain the actual active ingredient. Sometimes it's going to be a simple drug like a painkiller, and other times the placebo is going to be the best currently available drug for that specific disease. And that is done to ensure that patients are not deprived from getting medicated, so the placebo could be the best available drug. Results from each phase are going to be assessed to evaluate the beneficial effects against the harmful side effects, and then a decision can be made whether a license can be given or not. So this brings us to the end of this video. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.